Okay then. Okay. All right. So hello everyone. My great pleasure to welcome you to the Dean's Digital Cafe after a brief hiatus. So I'm William Letzu. I'm an assistant professor in the Biological and Chemical Sciences Department at New York Tech. On behalf of Gene Moore, I'm pleased to welcome you and I'd like to thank you all for all of New York Tech students, faculty, staff, and alumni for joining us here at today's event. So I'd like to remind everyone that the event is being recorded and we'll take questions following the presentation, but you should feel free to enter questions and comments into the chat at any time. And then also stay tuned for our new Dean's Digital Cafe page on the New York Tech website, where you can watch recordings of today's and previous events. So before introducing Dr. Tawfeek, I'd like to acknowledge the land. The Long Island campus of New York Tech sits on a large territory known as Pominoke, which was home to numerous indigenous communities, including the local tribes of the Matancox and the Merricks. I'd like to acknowledge the land, the stewardship, and sovereignty of Native peoples and their enduring importance, even in this virtual environment, to our working community here today. Okay, and now I'm pleased to introduce today's speaker, Dr. Hesham Tawfeek. Dr. Tawfeek recently joined New York Tech in the fall of 2024 as a research associate professor. Excuse me. He did his medical training in Egypt before moving to the US, where he joined the endocrine unit at Massachusetts General Hospital as a research assistant. He was a postdoctoral fellow at Harvard Medical School and an instructor at Emory University School of Medicine. Since 2011, he has worked at Columbia University in the Department of Orthopedics, the James J. Peters Veterans Affairs Medical Association Medical Center in the Bronx, and the Icahn School of Medicine at Mount Sinai. At New York Tech, Dr. Tawfeek now leads a translational bone research program and employs innovative pharmacologic and genetic approaches and animal models to understand the mechanisms behind bone loss associated with aging, hormonal imbalances, and immobilization. His work focuses on uncovering the role of bone and immune cell interactions in bone health and disease, with the aim of developing novel therapies for improving bone structure and reducing fractures in individuals with poor bone health. So Dr. Tawfeek has published in papers in many top endocrinology and physiology journals and sits on the boards of many others. Most recently, he was featured in New York Tech News for his receipt of a $1 million grant from the Department of Defense for studying spinal cord injury and the molecular level processes that contribute to spinal cord injury-induced bone loss. Well, with that, welcome, Hesham, and we're excited to learn more about your exciting work.
Thank you very much, William. And
yes.
Thank you, everyone, for joining.
Yes, we've got a very good audience now.
Yes. Yeah, I'm going to just share my screen again.
Not yet. Yeah. There we go. Almost there. Okay, we see your desktop. There.
You see it now?
Yep. And then put presentation mode and we'll be good to go.
Okay. It's good now? Everything? Full screen?
It's blue right now. I don't see the... There it is. There it is. Okay. All right. All yours.
So again, thank you, everyone, for joining this seminar. And this is my first seminar here at the NYIT. The topic of my talk today is about immobilization-induced bone loss after spinal cord injury, challenges and progress. So I would like to start with some general background, just to give you an idea about some concepts in bone. Then I will move after that to the actual talk. And I will try to use a very simple language and also try to make it more educational than really making it like a very basic talk. So contrary to what some may think, that bone is an inactive tissue, bone is actually a very dynamic tissue, and it undergoes continuous bone remodeling to maintain bone health and to repair micro cracks. And the process of bone remodeling actually involves two events in the bone tissue. One event is bone resorption. That's it's basically bone degradation at some sites of the bone tissue, and that's induced by osteoclasts, which are the bone resorbing or the bone degrading cells. And at the same time, after bone resorption by the osteoclast cells, then the bone forming cells or osteoblasts move in the space that's resorbed, and they start to form bone. So that's basically the process of bone remodeling. And the net of these two events is what leads to bone mass. So normally in a healthy skeleton or in someone who's in their 30s, for example, you would have more bone formation than bone resorption. So the net bone mass will be bone gain. So in contrary, in senile osteoporosis or in old age or in some other diseases of bone order, you will have more bone resorption than bone formation. And so the net bone mass will be bone loss. That's just an important concept to understand about bone before really we get into that talk. And since my talk is talking about immobilization and the paralysis and the interaction between these two problems and the bone loss, I will have to give a little bit of background about mechanical forces because that relates to mechanical forces in bone. So mechanical forces that are normally exerted on the bone by weight bearing or even by muscle contraction, they are very, very important for maintaining bone mass and preserving bone integrity. And these examples of this mechanical loading or weight bearing.
Turns your audio on.
Is everything okay?
You
Can you hear me?
no that
Hello?
Yes, we
Good.
hear you.
Yeah, we're
Okay.
good. Keep going, Hesham. Sorry about that.
Okay, that's okay. So some of the examples of this mechanical loading and weight bearing is simply standing. When you are standing, or even when you walk or exercise, there is weight bearing here that's exerted in the skeleton. And that's important because it stimulates the bone forming cells, the osteoblasts, which start to make more bone. And that's by promoting osteoblast progenitor cell recruitment and differentiation, and then making or synthesizing bone matrix. The osteoblast cells or their lineage cells, the osteocytes that are embedded inside the bone matrix, they are mechanosensors of bone. So they respond to any force exerted in the bone. And there are some molecular pathways at the level of the cell, including gap junction and Wnt beta-catenin signaling and prostaglandin E2 and even some other molecules are involved in transmitting that mechanical force at the level of the cell to translate that into bone formation or bone resorption. So what happens when there is mechanical unloading? So conditions causing some mechanical unloading, such as spinal cord injury, which I will talk about shortly, and also prolonged bed rest in some of the patients who are in bed for a long time for undergoing a major surgery or some other medical conditions. Also on space flight, where people are in the space flight with minimal weight bearing, or some muscle diseases that also impact muscle contraction, they can cause bone loss. So unloading itself can also cause death of some of the bone cells, and they can also recruit more osteoclasts, which are the bone degrading cells. So now I'm going to move to the actual talk, which is about the spinal cord injury. And the first part in the talk that I would like to share with you is about the challenges of a spinal cord injury bone loss. Why is bone loss with spinal cord injury is a big of a problem, especially in the clinical field? And I will share with you some findings, which many of them actually are facts, so that you become really aware of that problem. It is a very significant problem. So a spinal cord injury, as we know, when someone they injure their spine, which that's in a car accident or the fall, or there is a bullet or a stab wound, or even in the war as a result of an exclusion, it's a major insult to the body, and it impacts many systems of the body, and they become dysfunctional, especially if the injury is at higher level in the spinal cord. So as it relates to bone, these people who become spinal cord injured will be paralyzed partially or completely, again, depending on how significant or how complete the injury is. So there is loss of mechanical loading, and I told you, or just told you about the importance of having mechanical loading in our bones. So there is loss of mechanical loading. There are some hormonal imbalances, and there are some autonomic dysfunction. All of these actually impact the skeleton. Another problem or another factor that makes it more challenging, that almost every person who has a spinal cord injury and they become partially or completely paralyzed, they will suffer bone loss. Almost every person. So it's one of the most common and the severe complications of a spinal cord injury. And if the injury is complete, it's much worse when the injury is incomplete, because those who are completely paralyzed cannot move, like many of these people in wheelchairs who are completely paralyzed, they will have severe bone loss. And bone loss actually impacts all the sites of the skeleton below the lesion. And this affects mainly the long bones of the lower extremities. And the bone loss in these individuals, it starts very, very rapidly after a spinal cord injury. And it's very progressive, and it affects the trabecular part of the bone, which is the spongy part of the bone. And it affects also the cortical bone, which is the compact part of the bone. And I will show you a picture of that now, so you know what's trabecular and what's cortical. And again, bone loss is most severe in this individual, is most severe in the trabecular bone of the distal femur and the proximal tibia, basically the area around your knee joint. So above or below the knee joint is the area most affected in spinal cord injury, and it's the area also I'm going to talk about the fractures. The area, it's the most common site of fracture, and that's actually different from other diseases of bone loss like postmenopausal osteoporosis, which takes more the spine and other bones. One other fact is that, and again, look at these numbers, and you will really realize the problem. Bone loss and spinal cord injury is lost at a rate of up to 5% per month. And almost in the first year, these individuals lose about 41% of their bone mass. That's only in the first year. And it keeps going. And there are some literature that it never reaches a steady state. It keeps going. So they keep losing their bone, but most of the loss happens in the first year. And again, you see the statistics here, 5% of your bone mass is lost every month, and up to 41% is lost in the first year. How is that compared to other conditions of bone loss? If you look at microgravity, like again, I spoke about astronauts and space flight, and these people actually, they have a big problem in their bone loss, which when they come back from the space, it lingers around for some time. But again, it's still not as bad as spinal cord injury. Those also who are bedridden, they also suffer bone loss. But still, the magnitude of loss is not as severe as a spinal cord injury. There was at a rate of about 2% per month. And even in senile or postmenopausal osteoporosis, the loss is at the rate of 3% per year. That's not per month, that's per year. So you see how different it is. You see how severe the bone loss was a spinal cord injury. So, and again, when we now move to the fracture. So when you see bone loss, because some of us may think, well, these people are paralyzed, they may not really be moving around a lot, and even if they have bone loss, we are not going to worry too much about fracture. But no, they can fracture the bones up to 50% of spinal cord injured patients, and sometimes I will say SCI just for abbreviation. So spinal cord injury, it's abbreviated all the time, SCI, if I just forget of saying spinal cord injury. So 50% of SCI individuals they sustain a low impact fracture after injury. And the low impact fracture is actually a fracture from minor trauma. In fact, many of the spinal cord injured individuals, they have a spontaneous fracture. They don't even remember that they had any kind of trauma or falling or anything. Sometimes they may be moving from their wheelchair to the bed and they fracture their bones. So their bones are very, very fragile. And sometimes it doesn't even require really an actual trauma to fracture their bones. So these are all factors that make it very, very challenging. So even though it's their limited mobility, they still fracture their bones. So, and again, the most common fracture site is around the knee. And about 20% can have fracture around the hip. Another challenging problem is that bone loss with a spinal cord injury is very resistant to treatment. So when we talk about, for example, senile osteoporosis or postmenopausal osteoporosis, there are many drugs that are available and they are very, very effective actually in treating bone loss with a spinal cord injury. Some of these, one line is a line of drugs that inhibit bone resorption, bisphosphonates and some other medication. There is also a line of drugs that can stimulate bone formation. There are many of them like teriparatide, abaloparatide, or romosozumab, and they are in the market and they are very, very effective and they treat bone loss with aging. But when you try any of these drugs and spinal cord injured, they are very resistant. They don't respond to these agents. So there is a problem of responding even to treatment. And also when you try rehab like physical therapy, some exercising, some even mechanical stimulation or electrical stimulations of the muscle to stimulate some muscle contraction, they are very, very ineffective. So there is barely any response to all available pharmacological rehabilitation therapy. So strategies to preserve bone structure and prevent fracture after spinal cord injury are badly needed. And again, we really want to treat bone loss in these individuals so we can avoid the devastating complications of fracture. These patients, on top of their spinal cord injury and their other medical problems, they have a fracture. It's a big of a problem for these patients. Again, for their health, some of them may even die because of the fracture, and also the cost of that and for these patients. And also, when we ensure that we preserve the skeleton of these individuals, we can allow them a safer lifestyle. So these people, and you may have seen some of these people, they like to exercise, they like to move around. And again, with the improvement of healthcare and the technology, they are very much like normal people now. They go out and they play and exercise and do all of that. So, but if their skeleton is very, very weak, with any increase in this physical activity, they can easily have a fracture. But also improving their skeleton or preserving their skeleton is very important for these patients because they can use the robotic extraskeletal rewalk devices. And you may have seen some of these devices that people wear and they are completely paralyzed. But these devices, when they put on, they are automated in a way that they can help them very much train to walk. But if their bones are weak, they will easily fracture their bones. In fact, before they use these devices, they are actually screened to make sure that their bones are strong enough. And again, because many of them have weak bones, they may not be able to use this, and which again would make them very much walk around using these devices and become these devices can become more popular, and it can improve the quality of life for these individuals. So just to share with you a piece of data from our own data. This is a drug, abaloparatide, and abaloparatide is a bone anabolic drug. It's a drug that's in the market that was approved by the FDA for treatment of severe postmenopausal osteoporosis, and this was approved in 2017. And it's probably one of the most potent drugs in the market for treatment of osteoporosis. And we used this drug in mice that were spinal cord injured. And when you see, I mean, just quickly, when you look at this bar here, these are normal mice. This is how much bone you have in your femur. And the femur is the bone of the thigh. So we spinal cord injured these mice and we treated them with this drug, and then we sacrificed the mice and then we isolated their femurs. and we analyzed them by micro CT scanning, and I will tell you exactly now what micro CT scanning does, and it actually does give us an idea about bone structure and architecture and the bone volume. So when you look at the first bar here, these are normal mice that were not, they went through the surgery, but they were not spinal cord injured. So they are walking around fine. So this is how much bone you have in the normal mice that didn't have any treatment. So what about the mice that are spinal cord injured and are paralyzed in their hind limbs, which are the limbs of the mice that are in the back? These mice can walk around using the forelimbs, the front limbs, but their backbones will be paralyzed, more like people in wheelchairs. They lose about 40% of their bone mass, and this is an experiment for 5 weeks. So this is these mice, if you don't give them any treatment. When we give them this drug, it barely did anything in these mice. It didn't have any, they were very, very resistant. And that's again consistent to some of the clinical data. So you can see how challenging this problem is, and really the need for finding, understanding, and finding some good therapeutic targets in these individuals. So I'm going to talk now about the progress, and I'm not going to cover all the progress, but just roughly to give you an idea about studies that are not ours. Most of the studies, again, as I mentioned, they show that there is barely any response to the currently available medications for treatment of bone loss. But these are data, again, from our recent work. So, and I will tell you the story and how we got to this data. When I was working at the Bronx VA, and I became in charge of this project, we wanted to understand exactly what happens in the bones of spinal cord injured mice. We tried to see what may be compromised in these bones compared to the healthy bones. So we did some high throughput RNA-seq analysis, and this is analysis we take the bones, we flush everything in the middle, the bone marrow cells, immune cells, all of that's flushed out, and we just take the bones which has the bone cells in it, and it mainly has the osteoblast, osteocyte. They are enriched in these cells, and then we lysed these bones, and we did some gene analysis to see the difference between the gene expression in the normal mice versus the mice that were spinal cord injured. And what happened is that we came across some of the target genes, and we identified some of the transforming growth factor beta target genes. And the TGF-beta is an important cytokine. It is important in many functions in the cells and in the body, and also very, very important in bone. So we found that some of the genes that are target of that protein in the bone, and some of the genes that also modulate the function of that protein in the bone are compromised, or that the expression is altered in the spinal cord injured mice. So that's how the story started. And it was a very small pilot experiment, but we got an idea about where we can start. Then we looked into the bone marrow itself. We took bones from these paralyzed limbs like the femur, as I showed you, and we looked in the bone marrow about the level of that protein, TGF-beta1, and the signaling also, and we found that there is an increase in the level of that TGF-beta1. It's actually for bone, an increase of that protein is bad for bone. Just to make the story easy for you, when you have more than normal of that protein in the bone marrow or in your bones, it has bad effects on bone. It will cause more bone loss. So, and again, we know that altered the TGF-beta signaling contributes to other disorders of bone loss. So all of this evidence give us the rationale to really test two hypotheses here. The first one is that we hypothesized that inhibition of TGF-beta1 signaling will attenuate SSI bone loss. So if we have that protein and we have these target genes there, so have a good evidence that it may be at least one of the reasons why there is bad bone loss, or there is severe bone loss in these individuals, or at least here in these mice. So we said that, well, if we inhibit that signaling, then we will be able to prevent SSI-induced bone loss. And the second hypothesis is very similar, and I will explain the difference soon. We hypothesized that inhibition of TGF-beta signaling, if we inhibit that signaling, then we'll be able also to restore bone mass in these paralyzed mice. And again, I will talk about the difference between the two. So first, I will talk about the inhibition of TGF-beta signaling will prevent SSI-induced bone loss. So how did we do that experiment? Again, as I said, we have two groups of mice, male mice, and they are 16-week-old. And the reason why we use this age is because it's similar to the age in humans that get spinal cord injury. And these are male mice. Again, we started with males because spinal cord injury and the complications are more in the males. Males seem to have more spinal cord injury than females. It's a ratio of 4:1, actually. But we did also female studies, but we wanted to start with the male first. So we had two groups of mice. We had the sham group, which is very much the same group of mice that went through the surgery, had the anesthesia, we cut in the back and everything, but we don't damage the spine. But the spinal cord injury is the group that had the injury. And the injury, we do the injury by contusion spinal cord injury. We have an impactor that drops a metal probe in the back of the spine after we expose the spine, and it crushes the spine. And that's very much mimicking the real spinal cord injury in real life by bullet, by car accident, by falling, because there is another model of transection that people just open the spine and that's just slices very much by a blade. But contusion is more clinically relevant. So again, we have these two groups. Then we split them further. We say, okay, each group we are going to give them. One of them is a control antibody, an antibody that wouldn't really do anything on bone or in the body. We'll call it IgG. And then we have the anti-TGF-beta. We have an antibody drug called the 1D11 that interacts with that increase in TGF-beta and it inhibits its action. So we have an IgG group and they have a 1D11 or the blocker antibody or therapeutic antibody. And we give that antibody, these antibodies 10 milligram per kilogram per day. We give them twice a week. And we continue for 4 weeks before we sacrifice the mice to isolate the bones. And we analyze the bones by micro-CT for bone structure or histology to look at bone cells and the changes in bone formation and resorption. We also collect bone marrow elute, as I said, from the bone marrow of these paralyzed limbs, and we collect the serum also from the mice, and we do some protein analysis. So first, I will show you some of the micro-CT analysis data. Again, the micro-CT is a scanning of the bone, and what you see here, this is on the left. Can you see the pointer when I'm pointing? Can you see that?
Yep, we can see it.
Okay, so this is the femur, and this is a femur, actually, a whole femur scanned by micro-CT. And we very much focus on the area that's in the square here, because again, this is the area that's most impacted by spinal cord injury, where most of the loss is and where the fracture is. And we get parameters, and I'm not going to show images a lot. I'm going to talk about the actual numbers because that's this is just one mouse. But the numbers is really a group of mice, up to 8 mice per group. So you look at the statistics and you see the consistency of that. So we get also pictures like this is the cortical bone, 3D. And this is the trabecular bone or the sponge bone, where this is the most impacted part, is the sponge bone. This is where most of the loss is. There is loss in the cortical bone, but it's minimal. But the fracture also and the loss is most in the areas where you have more trabecular bone here. So again, we get a lot of parameters about bone volume, which does tell you that the whole bone volume. Then we get about data about the thickness of this trabeculae, these things here that look like noodles. That we get the idea about the thickness, about the separation, how separated they are, how connected they are, if they are more of noodle-like structures like this, or they are more of plate, more thick-like structures like this. And I will show some of these data. So again, this is the graph here on the left. This is the bone volume. don't worry about the fraction and all of this is basically just the bone volume. So these are normal mice that were treated with the control antibody. And these are mice that were spinal cord injured and were treated again with the control antibody. So this is how much, this is here about 50%, you lose about 50% of the bone volume. So what happens when you give these mice, you treat them with the therapeutic or the blocking antibody, which is 1D11. Let's not focus about the normal mice because that's not where we worry now. We worry about the spinal cord injured mice. So you look at the spinal cord injured mice, we managed to bring their bone volume from here, which you can look at that as not treated versus the treated here, almost more than twice. And you can even bring the bone volume to close or even a little higher than the normal mice. So that was really a great finding. And these data were very, very, very reproducible and they were very, very consistent. And you can look at the images here. And I got the 2D images because it's much easier to see with in the 2D than when you have the 3D. The 3D is a little crowded. So this is a normal mouse. This is when spinal cord injured. You can see through it more, meaning that there is less bone. This is when you treat them with the antibody. You very much bring the SCCI or the spinal cord injured mice to normal mice. If you look at this image here versus this image here. So, and we looked again at the thickness of the trabeculae, and we very much see the same thing. The antibody increases the thickness of this trabecularly after they went down about 30%. And again, these are all different parameters, but we very much rely on the bone volume because we want to see overall if the volume of the bone changed or not. These are also important, but we very much see most of the changes, or this is where we have a big window to really be confident about your data. So, excuse me. So some other parameters that we measured also are structural model index and connectivity. And the structural model index, if you remember in the first image I showed you, this does tell us about whether the structures there are nodal-like or rod-like or they are plate-like. And a lower number, a higher number is bad. So when you look at the normal mice, this is how much they have in this kind of structures. When we look at spinal cord injured, the number went up, meaning that there are more rod or nodal-like structures than plate-like structures, which means will be weaker bones. When you look at the spinal cord injury, there was an increase in this number compared, a decrease in this number compared to here, which is good. This is what we want. And we looked at connectivity, which means how these structures are connected together. And if you look at the spinal cord injured mice here, the connectivity is lower, much lower than the control mice. And when we give them the drug, we very much bring the connectivity. They become more connected together, similar to even the normal mice. So if you look at that, spinal cord injured with treatment versus the normal mice, they are very much the same. So almost brought this mice to normal. We looked again at cortical thickness, and just to make the story short for you here, spinal cord injury causes again about 20% or 15% decrease in cortical thickness. The antibody did not treat that. So it was a selective effect in the trabecular or the spongy part of the bone, which was more of our problem because again, most of the fractures happen at around the knee, the areas that are rich in the trabecular bone. So we were looking for something that will improve trabecular bone structure. We were not very concerned about cortical bone because it doesn't seem to be a site of concern. So we looked at bone histomorphometry, and I'm just going to go through a little bit faster here, and some of the data that are negative, I'm just going to flip through. So we looked at bone histomorphometry, and these are more like sections in the bone. We look at counts of bone forming cells, the osteoblasts and the osteoclasts, and we can do some calculation, get some statistics with that. And when we looked at the osteoblast number or osteoclast number, there wasn't really, first we looked at osteoblast number, and then we looked at osteoblast surface, and we didn't see much of a difference there. When we look at the osteoclast, the bone resorbing cells, we see a decrease actually in osteoclast number, which is a little surprising, and we do see a decrease in osteoclast surface, and I have some explanation for that. Again, when we look earlier, which we did before in some other experiments, we can see the changes that we expect, which is an increase in the bone degrading cells, osteoclasts, and decrease in the bone forming cells. But when we go a little longer, we very much miss the window where we can see the changes. So that's the simple explanation, but there are even other explanations of any of you would like to talk to me about that. But just for the sake of time, I have to move a little bit quickly. So we looked at, now we have the bone marrow elute or serum, which is the elute in the bone marrow of the bones in these paralyzed limbs. And we also have the sera that we collected from the blood. And we did some analysis, and we looked at markers of bone formation and markers of bone resorption. And again, very much we don't really see a change similar to what we see in the histology, and the explanation is very much the same. I told you, other than an increase even in bone formation in these mice that are paralyzed. And again, I have an explanation for that, which is what I told you before. But also, we do see sometimes that these mice, their osteoblasts at some point try to make bone, but there is a defect, it seems, in mineralization, so they can make probably some matrix. That's why we see an increase here in marker, and that's also has been seen in humans. You can see a marker of an increase of bone formation, but when you look at the bone, it's lost. So the cells are somehow adapting and trying to make some bone, but they cannot translate that even into completely fully mineralized bone because of probably a problem even in the osteocyte, which is the bone that complete the cells that complete the bone mineralization, and that's very important for bone mass and bone structure. So again, I'm going to skip through these. These are some of the findings, the striking findings that we see, which is here. This is the level of bone marrow of the TGF, that molecule I mentioned to you earlier, where we built all this story. So this is the level of this TGF beta 1 in the bone marrow and the bones of these paralyzed limbs. And you see here, this is a normal mouse. This is how much they should have in their bone marrow. This is in the spinal cord injured mice. They have almost twice. So when we treated them, as you may imagine, the antibody, it's a blocking or neutralizing antibody, it brought this down. And again, it did the same thing in the serum, which is total what we expect, because we injected these animals intraperitoneally, so the antibody gets everywhere in the body. And also, we will discuss that later, that antibody may also be improving some other function in the body other than bone, because again, as you see in the serum, also decreases the level of that TGF beta in the serum. Some of the other findings that we found in the bone marrow is that we saw an increase in the prostaglandin E2. Prostaglandin E2 is one of these molecules that do affect bone remodeling and the bone mass. Actually, when you have a persistent increase in prostaglandin E2, there is literature showing that this will cause bone loss. But if you give it at single shots, it may cause even maybe better for bone. But when it's persistently or chronically increased in the bone, it's bad for bone and it will cause bone loss. And this is what we see here. This is a normal mouse. This is how much prostaglandin you have in the bone marrow. This is the spinal cord injured mice. You almost increase that twice by having spinal cord injury and paralysis. And when we treated them with that blocking or therapeutic antibody, we very much bring that down. So that may be a mechanism also explaining how we prevented bone loss in these mice. We looked at some other factors also that are involved in bone, and again, bone marrow sclerostin and DKK1. These are inhibitors of Wnt signaling, and if they are increased, they are bad. But again, we didn't see changes, so I'm just going to skip through that. So we looked then at some. We looked at the molecular level, and the way to do that also, we took the whole bone and then we flushed the bone marrow cells. So we had the bone only, and then we grind that bone and then we analyze it for some of the proteins to see the difference and the effect, to see the difference between the control mice and then the spinal cord injured mice, and the mice that were treated with the antibody or were not treated with antibody, and to see how that alters some of the proteins in the or the molecules in the bone. So these are spinal cord injured mice, and this is an increase in a protein called SMAD2, and that's a downstream target inside the cell for TGF beta 1. When you have an increase in that TGF beta 1 protein, you will phosphorylate that these bands or these proteins inside the cell, and that's the signaling pathway. And this is what you have in this. These are normal mice. So the bones of these normal mice, the phosphorylation of that SMAD2, which again downstream target of that TGF beta. This is how much phosphorylation you would have in normal mice that walk around the normal. This is how much phosphorylation you have in their bones. But when you look at the spinal cord injured mice, there is massive increase in that phosphorylation. You see here how dark these bands are. And this is very much the total, the unphosphorylated or the total SMAD2, which did not change, which what we expected. So spinal cord injury causes a massive increase in phospho-SMAD2. And again, we expected that, and that's consistent with us that there is enhanced TGF beta signaling in these bones, which again is bad for bones. So what happens when we give them the blocking antibody? We very much neutralized that TGF beta 1, and we were able to very much inhibit that phosphorylation that we see here. So they look very much like the normal mice here. If you look at this and you look at this here. And this is very much a quantification. So you don't really have to look at bands or anything. We quantified the density of these bands, and we did statistical analysis, and that's what you see. These are normal mice, and these are mice that were spinal cord injured, and you see an increase in phosphorylation of that target protein inside the cell. When you treat this spinal cord injured mice with that blocking or therapeutic antibody, you see you bring that phosphorylation down from here. We're not treated or treated as control antibody down to here. We looked further into how that may be affecting the osteocyte cells, and the same thing, we took that lysate that's enriched in the osteocytes, which are the bone cells embedded in the bone, and we did some analysis on some of the cytoskeletal protein, and that's that's more molecular analysis. So we looked at a protein that's called tau, and that's a protein that affects the microtubule assembly. And if you have a lot of this protein or phosphorylation, more phosphorylation of that protein or a lot of it, it can cause a problem with microtubule assembly, and that can affect the cell function in many ways. So when we looked at the spinal cord injured mice, we have more of this protein, actually, more than the one in the normal mice. But when we treat them with that therapeutic antibody, we were able to bring down the expression of that protein, which may be better for the microtubule assembly and the cytoskeletal function of the osteocyte cells. We looked also at the phosphorylation of that protein, which is very known in the literature, that especially in the CNS field and Alzheimer's and all these neurodegenerative diseases, that when you have phosphorylation of that protein, it causes a lot of problems again in microtubule assembly. And there is a lot of phosphorylation in the spinal cord injured mice compared to the normal mice. And when we treated them with that antibody, you really diminish that phosphorylation, and these cells, you would imagine that will have normal or better microtubule assembly and normal function. And that's the quantification here very much. So I'm just going to take the last 5 minutes here so this is all shows that that treatment with that therapeutic antibody that blocks the effect of that TGF beta completely prevents bone loss. It's totally prevents bone loss and protects these mice against development of bone loss. So what happens now, if we take these same mice in the last experiment, and we let them completely lose bone, and after that, we give them the antibody, not give them the antibody in the beginning. So we'll give them the antibody right after their spinal cord injured. We are talking now about protection or prevention. But can that treatment restore bone mass or bone structure? That's the big, and actually it's a more important question, because those who have already bone loss and you try to treat them are even a lot more resistant than when you give them the treatment in the beginning. In fact, it's not a routine treatment to give spinal to start treatment of bone loss right after spinal cord injury. So most of these individuals come to you after a year or two of spinal cord injury, when they already have lost a lot of their bone mass and a lot of function of their bone cells have been compromised. So the big question is, which we call after when they come after a year or two, call that chronic spinal cord injury. So can we use that antibody and try to see if we will restore bone structure? In fact, it does, and it does it even very well. So we had the same experiment like we had before, but we made the mice lose bone, and then we give them the treatment after 5 weeks for 5 weeks. And then we looked at bone volume, and we looked at volumetric bone mineral density. And you look at the graph here, these are normal mice. These are spinal cord injured mice. They lost already about 50% of their bone volume. This is when you give these mice the therapeutic antibody. You very much, you go from spinal cord injured treated with control antibody here, down here, up to here. So again, it brings their bone volume very much close, or a little bit even higher than the control mice. So it does, this antibody was also has a high efficacy in restoring the bone structure after spinal cord injury. That's again was fantastic, really, when I got this piece of data, because the challenge which many people face is that you may have a drug that may work even to some extent in the acute stage right after the spinal cord injury, but we want something that treats chronic spinal cord bone loss after chronic spinal cord injury. And that's again what we see also with density. Bone density is down to here. When you give them the antibody, it goes from spinal cord injured mice down here up to here. So again, in summary, I would summarize that very quickly. Spinal cord injury has deleterious effects on the sublegional, trabecular, and the cortical bone structure. And spinal cord injury causes decrease in osteoclast numbers and increase in bone marrow TGF-beta1 and prostaglandin E2 levels, and the phosphorylation of phospho-tau, as I showed you in the last couple of slides. 1D11 treatment, which is the therapeutic antibody, protects against SI-induced bone loss, but not cortical bone loss. And that's the second part of the slide I just showed you, that 1D11 treatment restores bone structure after chronic spinal cord injury. And we believe that at least 1D11 treatment inhibits TGF-beta signaling and attenuates SSI-induced elevation in prostaglandin E2 and phospho-tau. So in conclusion, we first, inhibition of TGF-beta signaling again protects against disuse, trabecular bone loss with spinal cord injury. The differential, again, if any of you would wonder why there are differences, why did protect the trabecular or the spongy bone, but not in the cortical bone? Again, these are two structurally, so geometrically, they are different. And there are different forces that are exerted in these two components of the bone. There are also some cell niches, some cells that are in the cortical bone are a little bit different from the trabecular bone. So these are actually things that we are trying to figure out why it was selected in the trabecular but not the cortical bone, and trying to understand that. And again, the effects of TGF-beta inhibition are likely mediated by balancing bone remodeling, preventing prostaglandin E2 elevation, which I told you when prostaglandin E2 is persistently or chronically elevated, it can cause bone loss, and also the hyperphosphorylation of tau and the osteocyte microtubule destabilization. There are many things at the molecular and also at the translational level that we are interested in pursuing in this study, and one of them is studies of my current DOD funded project. So when I understand the role of TGF-beta and bone loss with chronic spinal cord injury, we need to do more than that, especially at the molecular level, and we also need to go even farther than 5 weeks. We may go for up to 10 weeks, and then we give them the treatment. We want to understand how TGF-beta restores trabecular bone structure after spinal cord injury, and also the role of sympathetic disruption and immune dysfunction in spinal cord injury-induced bone loss. And hopefully in the future, can do some study with Michael and study fracture and spinal cord injury and the role of TGF-beta. And that's the acknowledgement. And I will stop here, and I'm happy to take any questions.
Thanks so much, Hesham, for a great talk.
Thank you.
We have any questions from the audience?
Can I close the slides now?
Yeah, sure. Unless you need to refer to
people
them.
can see me.
Yeah. Well, I have a couple questions actually, if I may.
Okay.
Yeah. So it seems like if I understood it, that the primary effect of this drug is to reduce the osteocytes, so to reduce the bone resorption. So is that the typical means of bone loss after injuries and like spinal cord injuries, that it's an increase in bone resorption? Is there any loss in bone formation from the osteoblasts as well?
Yeah, yeah, and that's what I briefly mentioned. Thank you again for this question, because it I thought it will come up. When we look early in this spinal cord injured mice, we see severe decrease or loss of bone formation and an increase in bone resorption. So the two bad things happen early when you do spinal cord, when these mice become paralyzed. When you look at bone formation after 1 week of injury, for example, or 2 weeks, you look at bone formation is down. You look at bone resorption, it's up. So you have two bad things going on, that your bone is being eaten up, and the cells that are supposed to get in, move in, and make bone formation are not doing their job. It's down. And that's why you get severe bone loss. I'm not sure about, there is literature that antibody can actually increase bone formation and decrease bone resorption. I really think, I'm not sure if that's exact. I think things are a little more complex because again, we used like the drug that I showed earlier in the talk. It does increase bone formation. Actually, it's very, very potent. It does increase bone resorption a little bit, but it is a bone anabolic agent, but it didn't do much at all. So I really think the problem is more than just bone formation and the resorption. I think you need to restore that, but also you need to restore the mechanisms of bone mineralization and all of that. So you can really not just trying to make some matrix that's not mineralized. It's a lot more of a complex story with spinal cord injury. And I think that antibody overcomes many of these problems together. But the literature, for example, but we have not seen that, as I showed from the data, at least at the 5 weeks time point. If we looked at 1 week or 2 weeks, which I will be doing that in the DoD project, we will be seeing that it may have increased the bone formation and inhibited bone resorption.
I see. Great. Thank you.
Yeah.
Yeah, Michael.
Hesham, great presentation. Just a very brief question.
Yeah.
So, given that TGF-beta has effects on almost every tissue of the body,
Yes.
right? So have you looked at off-target effects, given the dramatic decrease of TGF-beta in the presence of the antibody in serum?
Yes, that's again a good question. We are aware of that, and I really do believe that it may have some off-target, not really off-target because it's a bad effect, because these mice again have other problems and have other problems maybe in the kidney, maybe in some problems in the liver, some metabolic even problems. They also may have immune dysfunction. Actually, TGF-beta is immunosuppressive, and that's how I built my DoD grant. So it may also be improving the immune suppression that happens with spinal cord injury. When we block that antibody, that may cause some recover the immune function. So I am looking into that, and we will be looking actually in other organs, because it is important to know that this antibody may turn out to be the one that's resolving the whole problem together. Because again, with all the resistance that's happening with other drugs, it does tell us that it's not just the bone loss because something happening only at the level of the bone, something in other organs is also going on wrong, and that's causing some resistance and making it even worse. Because again, it's a very, very severe problem. And again, if you look at other disorders of disuse or immobilization, again, as I said, in the space flight and the bedridden people, the problem here is a lot more severe. So it's a multifactorial problem, and we better keep that in mind when we are using that antibody. So it may turn out to be doing some other things in the body that may be helping really, because again, the effect of also seeing it preventing and also restoring bone structure was amazing to see that it did that.
Great. Yeah, just maybe to follow up on that quickly. I mean, you kind of mentioned that TGF beta has a lot of other roles, and it was known to be important in bone loss. But you know you probably saw. Did you fully decide that was the gene to go after from the RNA-seq data? Was that clear that was the one gene that was really different, or were there
well,
other genes?
I will tell you why I am someone who doesn't go with really small changes, and we got a whole some mitochondrial problems, oxidative stress, which I looked into that in some other studies, but I was never able to get good funding for that. And again, that could add to the problem. So there are some clusters that we got, but I looked at really at the genes that were probably the highly expressed genes. And these are the ones when I took them individually, and I looked at the ones that were very highly expressed, and I tried to find something in common. So that didn't come as a cluster actually in the RNA-seq analysis.
Okay.
In the heat map. That's something when I looked at the very highly expressed genes and did some literature search and tried to find something in common. And what I found in common that they were all either modulators of TGF-beta signaling or targets of TGF-beta signaling. And that's when we went ahead and said, well, let's measure TGF-beta and also see if signaling is really increased in these spinal cord injured mice.
Great. Henry, I think you have a question.
I do. Thank you. Hesham, very nice presentation. Thank you for all that. It's very impressive, the effects of the antibody on bone structure. But how does that translate into function, right? So I saw that the antibody increases the bone structure, even in the sham animals, right? So is that a good thing? Does that make them super animals that are now more protected from or less susceptible to injury? And in the ones that were injured and got restored, does that antibody treatment now makes them functionally look like a sham animal?
Well, we didn't really. I'm not sure if you are you talking about function in terms of activity and things like that, or
yeah, like the animals able to use their legs. I mean, their whole bodies again, or how does that work?
Well, we were not really interested a lot because there is a lot about using that antibody in normal mice. So we didn't really want to invest a lot of time in comparing the effect of the antibody in the normal mice that didn't have that treatment versus the one that had the treatment, because that has been shown. It's actually if you somehow lower TGF-beta signaling in mice, whether pharmacologically or using genetically modified mice, you will increase bone mass. But that's very much the case with all the bone agents that are in the market. Someone who doesn't have a bone problem, if you give them these drugs, it will increase their bone mass. So we didn't really invest a lot of time, but we will use it because we want to see the effect if there is anything that's very specific for spinal cord injury that happens in spinal cord injury but doesn't happen in the normal mice, which means it's related to immobilization. But so far we have not come across that, but we have always wanted, and we try to keep that even normal mice that have the therapeutic antibody, so we can have a good control of contrast of seeing what the antibody may do in the normal mice versus in the spinal cord injured mice. But we didn't really look in in terms of activity. Obviously, I can imagine that these normal mice that had the antibody, they will have more bone strength. They will be more resistant to fracture. There is no doubt that we will see that. But again, I was not trying to rediscover the wheel, because that again has been shown, and I didn't want to invest a lot in looking. But it could be interesting if we find something that the antibody does in the context of spinal cord injury, but it didn't do it in the normal mice, which again, something of interest to us, but it's not the focus. We always look compare the data and see if that will come up. And that's why also in the statistical analysis, we look at the interaction. There is an interaction, meaning that is that effect specific for the antibody for spinal cord injury, or that we will also see that in the wild type. There is a lot that we can really do it. It's sometimes we just have to keep things a little bit focused and how much we want to address. But thank you. That's also a great question if we can see that.
Very great. Well, I think we're a little bit over time, but that was a very interesting talk. We learned a lot about bones, and hopefully we'll learn some more in the future as well. So again, and also thank you, Hesham, and thank you all for attending today. Please join us again, actually, just again next week, when Professor Melissa DiMartino from Psychology and Counseling will be telling us about exercise intervention on middle school students' emotional well-being. So take care, everyone, and have a great rest of your day.
Thank you.
Thank you for your talk.
Thank you, everyone.
Right.
Thank you.
Bye.